So it's a pleasure to introduce the next of our new fellows, a colleague of my own, Professor Jian Li, who is Professor and NHMRC Principal Research Fellow, but head of the Laboratory of Antimicrobial Systems Pharmacology at the Biomedicine Discovery Institute at Monash University. Thank you, Jian.
Thanks for the kind introduction and also invitation for the talk. Good morning, everyone. I would like to acknowledge and pay respect to the traditional owner of the lands. I'm honored to be elected to the Academy, and I would like to start by thanking my nominators, Professor Ross Koppel, and supporters and my mentors over the years. So antibiotic has been a cornerstone modern medicine, and you can say, I know this figure is not really up to date, but we should see the peak for COVID, but it won't change the conclusion. Basically, over the last century, the decrease in mortality really due to the cure of infectious diseases. And in this process, antibiotics play a key role here, as everyone knows. But if without antibiotics, we expect probably 30 to 40 patients die due to infection. Based on this very famous document prepared by British government, by prediction, by 2050, there will be like 10 million people died due to infection. I have no intention to make cancer people feel bad, but this is figure certainly higher than the cancer deaths due to cancers. And this means every 3 seconds, there probably one person died, unfortunately. And I'm not sure why I stopped working now. Yeah, so in 2017, the WHO published a priority pathogen list, and all these 3 different types of gram-negative pathogens on the top, which urgently need new antibiotics. Why antibiotic got a problem? AMR got a problem. I think the major reason due to the economic model, and based on the figure, you can say for any type of drugs, the R&D cost around 500 to 1,000 million. But if you look at profit, anti-cancer drugs go to almost 10 billion, but antibiotics like minus 100 million. So that's a major problem. And the reason for this could be due to a few major factors. The first is the first one really due to high daily dose. I think the key thing. If you talk about daily dose, antibiotic can be used like a 10 gram a day, not like a couple hundred milligrams for anti-cancer drug. And also we have very high safety bar. For phase 1 clinical trial, we have to make sure the drug is safe. And then we need to go through very intensive clinical trials and very short treatment period, and antibiotic resistance caused another problem. So that's all explain why big pharmas left this field. So this is my research overview. I have 3 streams. The first one really about clinical translation, about antibiotic and phages therapies. And second, really about fundamental research on mechanisms of toxicity, resistance, and activity. And third stream really about drug discovery. This includes both antibiotics and phage therapy as well. So putting into a time scale, so since my PhD, I have been working on the clinical translation through collaborations. We did a few clinical trials, and we published the first dosing guideline for polymyxins. And during the time, I also work on the mechanisms of toxicity and also resistance and activity as well. And from 2008, we got 2 NIH grants and to continue our drug discovery program from a concept to clinical trials. So I will talk about this very briefly. And this is polymyxin structure. It got a very unique structure, got both positive charges and hydrophobic regions. And they are very important for their toxicity, activity, and drug disposition in vivo as well. So the reason I'm mentioning that is when we discover new antibiotics, we have to consider multiple structure-activity relationship. So I will mention shortly. So for the first stream, we did quite a few clinical trials. And before I started my PhD, there almost nothing regarding how we use this antibiotic, the type of antibiotics. So we did a few clinical trials, particularly 2 funded by NIH. And we also did a lot of animal work and in vitro work as well to figure out how we can best use polymyxins. And in 2019, we published this dosing guideline, which can tell clinicians for patients based on the patient renal function and on different conditions, which kind of dosage shall we give based on the PK/PD target. And during this clinical trial, we learned a lot of lessons. Polymyxin got multiple components, got a nephrotoxicity, really bad reputation, and also got a very narrow therapeutic window. That's a big problem. But on top of this, polymyxin really not active against intravenous administration after. They're not very active against the lung infection. That's a major issue in the clinic because the majority patients really got a pneumonia. So all these criteria are really important for the target product profile down the track for drug discovery. For the fundamental research, in addition to the single drug, single target molecules, we develop a computational approach. Use all molecular dynamic simulations. We got the experimental data from the membrane lipidomics, and then we developed this model and use a computational approach to see how polymyxin really interact with the membrane, with lipidi. We can calculate interaction energy and then build a relationship between the free energy and also the biological activities. And through this, we hope we can do our AI-driven drug discovery, which we're doing now. And in terms of antibody-resistant mechanism, this just give one example about polymyxin-dependent resistance. You can say the drug, the bug basically only grow in the presence of antibody, very unique and can be easily neglected in the clinic. So we did genomics and also membrane transcriptomics, membrane lipidomics, etc. And we figured out fatty acid and phospholipid metabolism really significantly perturbed. And we also realized LPS loss got dramatically remodeling of the outer membrane. And then we did membrane lipidomics. We found the negative charge PG significantly increased. And through computational approach, we found only when the PG increased and these positive charge polymyxin molecules can bound to the outer membrane surface and stabilize the outer membrane. And that explains why the polymyxin-dependent growth occurred. And this is dangerous in the clinic because very likely we can't really detect this kind of resistant isolate very easily in the clinical laboratory. And we also did lots of nephrotoxicity work, including intracellular concentration measurement and cell biology work. It activated multiple apoptotic pathways through the CRISPR genome screening, and we found potential channel also play a key role here, which open door to a new drug discovery stuff. And through this, we found lots of useful information like multiple structure activity relationship there and a very narrow chemical space, and the chemical synthesis is also very difficult. So to develop a drug, if you modify the structure, we can change a whole bunch of other characteristics, not as simple as we thought, just the increased activity. So actually for polymyxin, we can only touch a very tiny chemical space there. So very briefly for the drug discovery program, and we started from the concept, the single drug, single target SAR model, and we built a more complicated structure toxicity model and PK/PD/TD model through clinical data. And then we pulled through the whole drug discovery pipeline in my group. And fortunately, we funded by NIH, 2 large NIH grant, and we got this drug molecules went through phase 1. And then hopefully we can start phase 3. So very briefly, this is the fundamental approach we used. We optimize the activity, but also minimize the toxicity as well. So we started from each amino acid, and then we figure out the key position we need to modify, and then finally we balance the different modifications that came out with the molecules. We decrease toxicity and decrease the bonding to lung surfactant, increase their activity in the lung as well. So that's the whole story about this drug discovery program. And very exciting, we found this new drug also active against the polymyxin-resistant isolates because in the lung area, due to the PK advantages, we can get much higher drug exposure. And that really helped the great killing in the lung area compared to polymyxin B. So that's very exciting. And based on the omics study, we can see the very minimal perturbation in the kidney tubular cells, and that's supported also by a bunch of bioinformatic analysis through transcriptional regulator network. You can see the drug molecules really got very little toxicity. And to finalize these drug molecules, we got phase 1 clinical trials, single dose, got very clean results. And this is almost like 9 times higher, safer than the current use of polymyxin B. And through multiple doses in humans, we got very, very clean dose regimens. And hopefully we can push this to even like a higher dose in the patient. And we'll get into a phase 3 next year. And to summarize, I'm really glad my research can cover from the bed to bench and then go to commercialization. And one of the really exciting things is establish the whole antibiotic drug discovery pipeline in my lab from the very early stage to clinical trials. And I believe academics can play a leading role in the discovery of antibiotics. And being a fellow, I'm looking forward to expediting the commercialization and clinical translation of biomedical discoveries. Thank you for attention.
Questions? Any questions for Jian? All overwhelmed. I'm going to ask you one very general question. So obviously you've done all of this work in your lab. Let me ask you, how much money did this cost you?
Yeah, that's a great question. So basically from the very beginning till we got these drug molecules cost about, exclude clinical trial, cost about 10 million. That's over like 13 years. But on top of this, we did phase 1 through collaboration. That's another 20 million as well. So far got 30 million.